Hi, my name is Shalini, Doctor of PARTHIPAN, and today I would like to present about non-specificity of human mitochondrial DNA primers towards non-human samples for allele-specific PCR approach. Forensic non-human DNA contamination in human biological samples often encountered by forensic scientists at crime scenes. Human samples are exposed to various non-human DNAs from surrounding environment, causing sample misses. These can produce false positive results during sequencing in human identification. There are few circumstances of sample mixtures between human and non-human can occur as shown in the picture below. The black tiny thing in this picture shows that there are ants found on human skin lesions where the necrophagous ants are eating the human skin tissues. Second picture shows larval colonization on decomposed dead bodies. Third picture shows that microorganisms which are such as bacteria are too common found on human skin. Furthermore, humans and pets such as cats and dogs will live first on human bodies. These circumstances could lead to sample mixtures between human and non-human samples. Therefore, it is important to specifically identify human DNA from non-human sample mixtures in forensic human identification. In this study, we used allele-specific PCR to detect point mutations in humans because it is rapid and cost-effective cycling method. We used mitochondrial DNA because it is stable structure resistant towards degradation, high copy number per cell, high mutation rate, and small size genome will add advantage to forensic applications. Single nucleotide polymorphism is most abundant found in human genomes and it has high throughput analysis for allele-specific PCR. Therefore, the SNP can be detected directly in cell electrophoresis without the need for sequencing. For human identification in this study, we chose SNPs that are seen high frequency among populations. Therefore, it is important to validate the selected human mitochondrial DNA primers with non-human samples to avoid false interpretation with human samples. Therefore, in this study, we used three classes of non-human samples from the category of bacteria, mammals, and insects. Bacteria are too common found on human skin, therefore we randomly chose E. coli from gram-negative group and Staphylococcus aureus from gram-positive group. In the category of mammals, humans are also categorized as mammals. Therefore, dogs, cats, and monkeys have genetic similarity more than 90% when compared with humans. Therefore, we chose dog Canis lupus familiaris, Macaca fascicularis, which is macaw, Felis catus is the scientific name of cat. We chose these three species under the mammals category to be validated using human mtDNA primers. And for the insect categories, we use Spinifera genitaliis species from fly and Formicidae, which is scientific name of ants, has been used to validate because these two species are found in colonization and feeding on dead bodies, and sometimes they are also necrophages, which will eat the skin tissues of human dead bodies. Moving to methodology, the process starts with primer designing. The human mtDNA primers are designed using NCBI primer blast that is specific to human mtDNA genome. These primers will generate templates that contain the SNP of interest to be used in allele-specific PCR. The parameters of the primers for product size is set in between the 250 to 450 base pairs. Second process is sample collection and DNA extraction. Non-human samples were obtained from relevant authorities from CITEM UKM, Wildlife Forensic, and Forensic Entomology UKM. The collected samples were extracted using DNA extraction kits, then run through the thermocycler for PCR amplification together with the human mtDNA control, which is called as 9947A, or also can be called as forensic human mitochondrial DNA. After the amplification, the product is confirmed by gel electrophoresis and visualized under UV transilluminated. Moving to results and discussion, there were 12 sets of human mtDNA primers were optimized as according to initial handling temperature as tabulated here. 25 microliter of total per reaction were used with the thermocycling condition were indicated as in the graph. The optimized human mtDNA primers were validated using the non-human samples as explained before. The result shows that non-specific binding of human mtDNA primers towards E. coli, Asaurus, dog, and cat samples occurs. The amplification of these samples were compared with human mtDNA control. This picture shows that E. coli produces one non-specific binding with the primer sets of fragment J and three DNA bands non-specifically produced by E. coli with the primer sets of fragment K. All the non-specific amplifications that occur during the PCR amplification with non-human samples are indicated as in the red circle. This non-specific amplification shows that the desired human mtDNA primers are not specific to human samples only, even though the designed primers are used to be built using the entire primer blast that is specific to human mtDNA genome only. There are no definitive studies prove that why this non-specific amplification occurs, but our findings suggested that the slight modification in annealing temperature, which is plus minus 0.1 to 0.2 degrees Celsius, can remove the non-specific amplification, as shown in the table here. The non-specificity of human mtDNA primers for E. coli, Asaurus, dog and cat sample has been increased from 83.3% to 100%. And this has been proved by the gel picture here, which shows the amplification only occurs in human samples, and the modification in annealing temperature has refracted the amplification in non-human samples. Finally, the modification in PA removed the non-specific binding of human mtDNA primers with non-human samples without the sophisticated re-optimization. Therefore, we do not need to remove the primer or exclude the primer from the experiment if it binds to the non-human samples. With the modification of annealing temperature, we can increase the specificity of human mtDNA primers towards the human samples only without binding with the non-human samples. Thank you.